I am going to walk you through how to make your standards and create a 4-point calibration curve to determine the percent copper in an unknown sample. For this portion of the experiment, you will need 4 test tubes, 2 different volumetric pipettes, one for water and the other for your copper nitrate, some distilled water, some 0.4 molar copper nitrate stock solution, and cuvettes to get absorption measurements of your standards. Here I am diluting the 0.4 molar stock solution into 3 additional standards for the calibration curve. The 4 standards are 0.4, 0.2, 0.1, and 0.05 molar. After the standards are made, they will need to be carefully transferred into the cuvettes. First, you will need to make sure you pour DI water in the first cuvette as a blank sample. Later, make sure to orient the sample into the sample cell of the colorimeter with the clear side in the path for measurement. It will align with an arrow. When filling the cuvettes, make sure to fill them most of the way. During this process, you can also wipe the clear sides of any smudges using a Kim wipe to ensure proper readings later. When finished, you see a nice gradient of blue, characteristic of copper nitrate in aqueous solution. These samples must be measured using the proper wavelength of the color of light that is absorbed, not transmitted by the sample. In this case, it is 635 nanometers. Zero out the colorimeter using the blank sample. You can then measure the blank, and the result should be 0 absorbance. Measure all of the standard samples. Here are the results that will be used to make the calibration curve in Excel later. Okay, time to prepare the unknown brass sample for measurement. Make sure that you have weighed and recorded the sample weight prior to dissolving the brass. Zero out the scale, then weigh the brass. Wait until the weight stabilizes before recording. To dissolve the brass, we will use 6 milliliters of concentrated nitric acid. The reaction will start, and the beaker may be warm. Place a watch glass over the beaker, then place the beaker on the hot plate at a very low setting. This will help speed up the reaction. Let the reaction continue until completion. This may take about 10 to 20 minutes. For this video, it was left overnight due to the fume hood being shut off unexpectedly. At least now it is the bright blue color we need. Normally here we add 30 milliliters of water and stir to help complete and cool the reaction mixture. Now the solution will be carefully and quantitatively transferred into a 100 milliliter volumetric flask. Rinse the beaker 3 times to transfer any remaining sample liquid. Fill up the flask with DI water most of the way with some room before the fill line. The solution meniscus will sit right on the line when filled. We will slowly add water until we reach the fill line so we don't go over. Invert the sample a few times to mix. Okay, now we can measure the sample in the colorimeter. Transfer the sample into a cuvette and wipe with a Kim wipe. Because this is a different day, I had to re-zero the instrument. It is better if they are run on the same day. Once you have your sample absorbance measurement, along with your standard measurements for your calibration curve, you are done here and ready for analysis of the data.